Annenberg Media.
By the National Science Foundation, America's investment in the future. By the American Society for Microbiology. And by the Office of Biological and Environmental Research of the United States Department of Energy. Additional funding was provided by the Arthur Vining Davis Foundations. This program has been made possible in part by the Corporation for Public Broadcasting.
As different as all organisms are, multicellular or single-celled, they all share similar chemical molecules like DNA. It is the DNA which determines how each performs a fundamental process, making and using energy. In other words, how they metabolize. How do organisms survive in this fickle and competitive universe? Well, for one thing, we all need food. Chemicals we can break down and use as energy to build new cells. This apple works fine for me, but it wouldn't satisfy a hungry carnivore. Different organisms, different energy needs. No matter what the habitat a microorganism may call home, all microbes are performing this one basic life process. A process we all must perform to survive and grow. Metabolism. Metabolism refers to the thousands of chemical reactions and physical workings of the cell. It is the flow of matter and energy. I'm Jim Leinfelder. In this episode of Unseen Life on Earth, we'll explore a variety of energy-producing metabolic strategies. Strategies determined by the molecules shared by all organisms, DNA, the backbone of diversity, protein synthesis, metabolism, and life itself. Cells are like chemical factories. They spend a lot of their time producing energy as ATP, adenosine triphosphate, the energy storage material of all life on Earth. They then use that energy to drive their chemical reactions. These chemical reactions are divided into two groups, catabolism and anabolism. Catabolism is the process of breaking down macromolecules into their smaller component parts. These components may either be further metabolized for energy or used as building blocks for making new macromolecules. This process of using the energy and simple building blocks to make new macromolecules for the cell is called anabolism. It is an organism's environment and the energy sources available in that environment that determine which energy-producing strategy is successful, and therefore which organism will survive. Organisms can obtain their energy in one of several ways, such as photosynthesis, the conversion of light energy into ATP, or chemolithotrophy, converting inorganic chemical energy, such as hydrogen gas or sulfur, into ATP, or fermentation and respiration, the conversion of organic chemical energy, such as organic waste or sugars, into ATP. They are different processes, but all can go on in the same environment, and some even in the same organism, at different times or under different circumstances. The way that life works is that primary producers, such as photosynthetic and chemolithotrophic organisms, use part of the ATP they produce to create carbon-rich compounds. Other organisms, consumers, break apart the bonds in these carbon-rich compounds to release the energy that has been stored. Primary producers, such as plants, form carbon-rich bonds, and consumers, such as animals and microorganisms, break them. It is one big cycle. Microbes, since they have been here longer, have evolved more ways to do it. In most of the world outside the laboratory, ecosystems are complex mixtures of organisms with different metabolic abilities that not only coexist, but function together to be more than the sum of the parts. Like this ecosystem, a wastewater treatment plant in Radford, Virginia. Despite all the hardware, it's metabolism-driven. Waste materials coming into a wastewater treatment plant come from a vast array of people who eat different things and have different microbes and therefore excrete different things, as well as anything else that goes down the sink or toilet. Throughout this plant, there's a frenzy of metabolic activity, but we're really here to focus on one, respiration. And to help me with that is Dr. Nancy Love, a professor of civil engineering at nearby Virginia Tech, and you're really here to talk
activated sludge. I love to talk about activated sludge.
Hey, who doesn't?
But before we talk about that, that's further down in the process. We need to spend a minute talking about this.
Okay, what goes on here in this big, pond, basically?
Well, the wastewater that comes in from the community, as well as some industrial inputs, comes in here, and you have to separate the liquid from the solids. And at this point, all we're doing is removing large solid particles from the liquid. And then the liquid is what we're going to pass on down to the activated sludge process, where the metabolism is occurring.
Oh, this isn't where the microbial action is.
Exactly. But do you want to go see that?
Yeah, let's get to the metabolism. Dr. Love, when my layman's ears hears the word respiration, I immediately start thinking about breathing, but that's not exactly what we're talking about here.
That's right. I mean, the organisms are breathing in a sense, but it's more complicated than that. The wastewater that comes into this plant has energy in it in the form of electrons, and the bacteria convert those electrons into a form they can use. It's a food source for them, it's an energy source. These compounds in the wastewater enter into the cells, the bacterial cells themselves, and when that happens, the available electrons have to go somewhere, and in this case, those electrons go to oxygen. So the organisms park the electron from the food source, the wastewater, on oxygen, and in the process of doing that, create ATP. That process of taking the electron from the wastewater compounds, parking it on oxygen, and producing ATP for energy, that process is respiration, and it is a form of catabolism. At the same time, not all of the compounds in the wastewater go through catabolism. Some of them go through another pathway and create body parts or cell materials to form new cells. So what happens is you have wastewater coming in with available energy, and it ends up forming water from the respiration process on the catabolic side and cells on the anabolic side.
And it all has to take place in the presence of oxygen, and the more oxygen, the more readily and rapidly it will take place.
Absolutely. There's a lot more energy coming into the system than the atmosphere has oxygen to satisfy. So it's very important to provide a lot of air, and that's what you see here, the swirling in the basin and swirling through the reactor, so there's plenty of oxygen available for all the organisms.
The mixture of wastewater and bacteria, which engineers call mixed liquor, is then brought to this clarifier, where the millions of bacteria and other particles settle.
The liquid goes through a few more processes and then into the river, but the solids go to one of two directions. Most of the solids, these bacteria that we created before, go back to the front of the aeration basin so that we can start the process of metabolism all over again. But if you think about what we talked about up there, the wastewater coming in enables the growth of more bacteria. And if we don't waste or remove some of those bacteria, we'd be up to our ears in bacteria in no time. So it's very important to remove as many bacteria as you make in one day. We waste what we make. So another fraction of the solids at the bottom of this clarifier are taken off to another side of the plant where we handle the solids. That's called the waste.
The solids to be wasted are transferred to an anaerobic digester. Within the anaerobic digester, a diverse community of bacteria perform complex fermentation reactions, which further reduce the volume of the waste, producing organic acids in the process. Other bacteria, called methanogens, convert the organic acids to methane gas. So, the anaerobic digester reduces the volume of the waste by converting solids to gases, enough gas to lift the tank's 125-ton lid.
So Dr. Love, I take it we've finally reached the end stage of this entire process here.
That's right. What you have here are the residual bacterial cells. Remember, back up at the earlier part of the bioreactor, we had solids that we said we had to waste. We waste what we make. And those solids went through several different processes where we removed water, we anaerobically digested the solids, where we killed the bacteria and we killed any pathogens that were present in the solid material. We removed some more water, and then this is what you have.
Essentially what we've got here are the corpses of all those bacteria.
That's right. They're corpses, but there's a very good nutrient value associated with this material. This material is actually applied to agricultural lands.
Many different nutrients remain in water emerging from a wastewater facility. Primary producers need such minerals to start the carbon fixation process over again. For the most part, this process is driven by the sun's energy, light. But what happens when there is no light? No problem. In the late 1970s, scientists in an improved deep ocean submersible were able to explore much deeper than ever before. At that time, it was believed that no life existed in the deep ocean. Over 2 miles down, scientists found underwater volcanoes spewing superheated minerals from the Earth's core. When these minerals meet the ice-cold water, they crystallize, creating huge undersea structures. One is 15 stories high. Spotlights pierce the blackness. There is no sunlight. Yet life blooms in the darkness. How? Microbes. Instead of sunlight, bacteria use the sulfur from the volcanic vents as the basis of their energy metabolism. These organisms are called chemolithotrophs. They convert inorganic chemical energy into ATP. The bacteria become food for some of the larger creatures and form the base of the food web. Tube worms, shrimp, fish, and crabs thrive in this oasis of heat and microbes. Much of this is still a mystery, but one thing is conclusive. These chemical-powered bacteria are at the center of this deep-sea food web, and the darkness of the ocean floor is not their only domain. The peaceful emerald forests and dynamic pools of the Pacific Northwest Cascade Mountains, a haven for photosynthesis. Business as usual for the microbes that flourish here. But chemolithotrophs, not entirely unlike those found beneath the surface of the sea, have been present over billions of years, waiting for an instantaneous, cataclysmic change in the environment that will give them their chance to metabolize and grow. In May of 1980, Mount St. Helens erupted, changing the surrounding environment drastically. The balance was tipped, and suddenly sulfur became abundant. The small population of organisms that metabolize sulfur suddenly had the opportunity of a lifetime, the opportunity to take hold and flourish, and they took advantage of it. Environments change, DNA changes, new niches are filled, and metabolism goes on. Some things change slowly, and other things change rapidly. In changed environments, organisms adapt to have a better chance of coping, surviving, and possibly prospering. Mount St. Helens and ocean sea vents are extreme habitats, at least from the perspective of a human being. But home is home to a microbe, anywhere food can be found. Another type of metabolism is one we human beings have gotten pretty good at using for our own purposes: fermentation. Baking bread, making cheese, and brewing beer. Very different habitats and systems, but the metabolic process is similar. People have been enjoying bread, cheese, and beer for thousands of years. However, early bakers, cheesemakers, and brewers really had no idea why yeast made bread rise, or formed holes in Swiss cheese, or turned hops and grains into brew. What they were doing was tapping the power of the microbial world, harnessing the metabolism of microbes, and enjoying the products of microbial fermentation. Fermentation is one of the oldest metabolic processes on Earth. It is a simple metabolic strategy in which organic compounds are broken down in the absence of oxygen into simpler organic compounds. In the case of bread, beer, and Swiss cheese, different organisms are involved to produce each product. But in each case, the organism uses fermentation. Simple compounds, such as sugars in the grains and lactose in milk, are broken down into simpler organic compounds like alcohol and CO2. The CO2 is what makes the bread rise, the beer fizz, and the holes in Swiss cheese. And whether it's cheese, bread, or beer, what's happening in addition to fermentation is growth. And as far as the microbes are concerned, growth means reproduction. No matter how big the vats, the process all happens at the microbial level, one cell at a time. And it has to be the right cell. Jamie Emerson is a brewmaster in Hood River, Oregon. For him and every brewer in the world, brewing starts in the lab. Jamie Emerson, Jim Leinfelder.
Hi, nice to meet you.
Nice to meet you. What are you looking at in the petri dish there?
Well, this is brewer's yeast. It's our favorite microorganism around here. What we've done here is used a plate and separated out single colonies of yeast, and you can see them as single dots here right on the media. And this media is basically provides growth material for the yeast to grow on, and then we can look at these cells individually.
So those are good results then?
Good results. If you would have seen anything that we didn't want on this plate, it would look blue because the media would give it a dye and colors it.
Okay. And so when you see one yeast isolated there, then you know that you can put these guys to work for you.
Absolutely. We can take a single cell here and begin growing it and actually use it to pitch a large fermentation tank with trillions of cells.
Wow. And here we've got, what, maybe?
Oh, a couple thousand colonies of yeast, give or take. Then with all these plates behind us, we use that to track cleanliness of the brewery, because basically brewing is 99% cleaning and 1% art.
Right.
And we use those back in an aerobic and anaerobic chamber behind you. So this is where we keep track of what's going on in the brewery from an aerobic standpoint on one side and anaerobic on the other. That way we can keep track of both kinds of ferments. Ours in particular are anaerobic. That's what we're mostly interested in.
So essentially you're basically a yeast farmer.
Yes, I am. I'm a yeast farmer, and here we're just checking that we're farming right, that everything is right, very clean. And you know, it takes a lot more than just what's in these petri dishes to make beer. And I'd happy to take you down and show you the farm.
Oh, great. Love to see it.
Well, Jim, welcome to the back 40.
I guess this is where the farm analogy starts to break down, but this is where you're growing more yeast and also starting the fermentation process?
Absolutely. You remember in the lab, we had the plate with just a few cells on it. We take a little bit of those cells and add it to the wort, which is unfermented beer. It's full of sugar. The yeast eat the sugar up through metabolism, making energy, more yeast. We keep adding more and more liquid in this tank till it's absolutely full. And in that time, the yeast are chewing up all the sugars, growing more yeast. And we're talking in the range now of trillions of cells in this tank. And then this tank is then pumped into one of the large fermenters, and we fill this fermenter all the way up. Okay. And by the time that process is finished, we're talking 7 quadrillion cells.
Jamie, how do you know that these 7 quadrillion yeast cells are metabolizing the way you want them to metabolize?
Well, besides the information we get from the lab, we actually take it out and look at it. Well, here you can see what's actually going on inside the fermenter. This is beer that's undergoing ferment. The liquid we ran in is absolutely clear, and you can see the haze in there, and that is the yeast cells that are multiplying and chewing up the food in there. You can see the bubbles coming up to the surface, and that's a byproduct of the fermentation. Yeast is metabolizing the sugar and making CO2 and alcohol.
As fermentation continues, the CO2 is vented off to prevent the increased pressure from killing the yeast and prematurely stopping the desired fermentation. The yeast cells continue to grow and grow to reproduce. The growth of yeast cells, as well as bacteria, can be expressed in a growth curve. The number of cells is plotted against a timeline. In the initial or lag phase, the population remains static as the organisms get used to their new environment. Next, in the exponential phase, cell growth increases exponentially, doubling with each generation, until the species reaches its metabolic peak. The peak soon becomes a plateau in the stationary phase, as the rate of growth or reproduction equals the rate of death. Death occurs for a variety of reasons. An essential nutrient may run out, waste could reach toxic levels, or a predator may appear. Whatever the reason, the population then enters its death or decline phase, and the organisms rapidly die off.
Once the yeast are done eating all the sugar, we can take them out of the tank, and here's the granddaddy of them all.
Oh, how do you know when they've finished eating?
Well, we only give them so much sugar, so when the sugar's gone, they're done.
Ah, okay.
Well, this is the culmination of our process here. Collecting some yeast.
Ew, so that's what's coming out of the bottom here?
You got it. And it may look like quite a bit. You know, there's several thousand million yeast cells probably in here.
Yeah, it looks like a malted milkshake.
But it's all one kind of yeast, all genetically identical, which is the trick with fermentation. There's lots of things to ferment, but we're only looking for one thing.
So this is a monoculture.
Monoculture.
I mean, talk about reproduction equaling growth. You started with a vial, and you end up with truckloads of this stuff.
That's right, and you want to see some growth just out of one tank.
Wow.
Gallons and gallons and gallons of microbes.
As the environment changes, new niches arise, and the microbes change to fill them. Over time, the number of niches expands, and so do the different kinds of microbes. As they develop new abilities, some of them keep all or part of the old information in their DNA, and that lets them respond if conditions change again. As different as these organisms are, and as varied the means of metabolism, all living organisms have the same goals: to make and use energy. The instructions for new generations and all the metabolic activities for all forms of life on planet Earth are passed along through DNA. It is this amazing molecule that we will focus on in the next episode of Unseen Life on Earth.
Annenberg Media.
By the National Science Foundation, America's investment in the future. By the American Society for Microbiology. And by the Office of Biological and Environmental Research of the United States Department of Energy. Additional funding was provided by the Arthur Vining Davis Foundations. This program has been made possible in part by the Corporation for Public Broadcasting.
For information about this and other Annenberg Media programs, call 1-800-LEARNER and visit us at www.learner.org.